My name is Paulina Guzman Tinoco, and throughout these last 9 weeks, I've been investigating filamentous algal blooms in mountain lakes, specifically with the OLEX lab. So why study mountain lakes? This is a picture of the lake in which I was studying, and these lakes provide clean water to downstream communities and wildlife. So all of us drink water, or we should be drinking water, and so that's one of the main reasons why water is so important. And my second reason would be because some of us just deal with the recreational value of lakes. So I personally, I like to paddleboard and things of that sort. A lot of people like to swim or just recreational values of these lakes. And so if we don't protect our water, that decreases the recreational value of our lakes. And mountain lakes are very isolated. They're known to be very nutrient poor, and they're also very sensitive to disturbances in the environment. I'm specifically studying filamentous algal blooms, and so I will have Emma pass around 2 vials of samples of filamentous algal bloom that I took out in the field. So filamentous algal bloom is thick hair-like mats of green algae that grow in shallow lake areas. So right here, I have me holding a fab. Right here, I also have me holding a fab, and this is a fab out in its natural habitat. And unlike regular planktonic algae, fabs are attached to the lake bottom, which is known as the benthic zone. And so, as I said, planktonic algae tends to float, but this just attaches to things like rocks. And so you can see I picked this piece of algae from that rock that's right next to it. And they thrive in nutrient-rich conditions, and they disrupt lake ecosystems. Fabs can also signal water quality changes, and they have become much more common in previously low nutrient lakes. So you might be wondering why we should care about filamentous algal blooms. So their sudden appearance suggests that something in the environment may be changing. So they are suggesting that something in the environment may be changing. So understand what drives these blooms can help us understand how we can protect alpine ecosystems and how can we guide conservation efforts and protect our water. And this is a map of Sky Pond, and this is a picture that I took of Sky Pond. And Sky Pond was my field site, and it is about 10,900 feet above sea level, and it is located in the Rocky Mountain National Park. So I came up with 2 research questions. The first one being, are concentrations of ammonia and phosphate higher in areas with fabs compared to areas without fabs? So ammonia and phosphate being my 2 nutrients that I was studying. And my second question is the amount of particulate organic matter, also known as POM, higher in areas with fabs compared to areas without fabs, specifically looking at Sky Pond? So why nitrogen and phosphorus? They are key nutrients that support algal growth, and these 2 nutrients are known to be very limiting nutrients in lakes. Increases in nitrogen as well as phosphorus can come from human-caused changes, so deposition, fertilizer runoff, and things of that sort. So it tells us a lot about climate change and how climate is just increasing. And as I mentioned, I looked at particulate organic matter, known as POM. POM, particulate means very little. Organic is recently dead or living. Matter is things, so if you put it all together, it's little, recently dead or living things. So tiny pieces of organic debris in the water, so think dead algae. This is dead algae in one of our filters. Plant bits, so pieces of leaf litter and things of that sort. And organic material can tell us a lot. It can tell us how much organic material there is in bloom versus non-blooming sites. So this was a water sample that I took from right above a fab site. And as I said, this is algae in a filter. Going on to my method specifically for field sampling and lab analysis, we had three little different types of methods. So field, lab, and analysis. So for the field, we would wake up really, really early. I'm talking 3 in the morning. Get to the Rocky Mountains at about 5-ish, and then we would hike up to Sky Pond to take water samples. So this is a picture of what our samples look like. So we would just label pieces of tape, put them on bottles, and then fill them with water. Then we would go out into the lab and filter our water through already like pre-weighed filters to collect organic matter. And then we would dry those filters, weigh them, ash those filters, and then weigh them again. And this was to measure organic as well as inorganic material. And then we would measure concentrations of nitrogen, which is a form of ammonia. And phosphorus, orthophosphate, which is a form of phosphorus, on a nutrient analyzer, which we just called the seal. And then we would analyze them using RStudio, and we ran statistical tests, which were t-tests, and it was my first time using R. So for sampling locations, I included the map again of Sky Pond. So we collected water from the inlet, the outlet, Site 1, Site 2, and Site 3, and our lake surface. So inlet is where the water comes in from. Outlet is where the water comes out from. Site 1 does not have fabs. Site 2 has fabs. Site 3 does not have fabs. And lake surface is exactly what it sounds like. It's just the surface of the lake. So this variety allowed us to compare bloom and non-bloom conditions, as well as shore sites and non-shore sites. Right now you're looking at my R graph. So this is orthophosphate by site. We can see that the water comes in with a high concentration of orthophosphate, and as the water continues to spread through into the lake, it kind of lowers. We didn't collect from the sample because we got caught in a storm. And then going into our fab site, which is shore site 2, we saw a huge increase in our nutrient orthophosphate, and then we could compare it to our non-fab site, which has extremely significant lower concentrations of orthophosphate. And so after running our t-tests, we found that there is a statistically significant difference between orthophosphate concentrations. Again, orthophosphate is our nutrient in our fab site, which is shore site 2, and all other sites without fab, so shore 1 and 3. And we also found that there is a statistically significant difference between orthophosphate concentrations in shore site 2 and all other shore sites without fabs. Now looking at our next nutrient, we're looking at ammonia by site, and this one's a little different. So it comes in with a low amount, water comes in with a low amount of ammonia, and then lake surface is a little higher. Keep going into the outlet, it's a lot lower than it was when it initially came in, and then a huge increase in shore site 2, which again is our fab site. And so after running our t-test, we found that there is a statistically significant difference between ammonia concentrations, our nutrient, between shore site 2 and all other shore sites without fabs. And we also found that there is a statistically significant difference in ammonia concentrations at fab site versus non-fab shore sites. Now looking at our POM concentrations, so we're seeing that there's not too much particulate organic matter, and then again, huge increase in our fab site. So after running our t-test, it's surprising because to the naked eye, it looks like a huge jump. We found that there's no statistically significant difference in particulate organic matter concentrations between shore sites and non-shore sites. So what does our nutrient data show? This is us hiking down from Sky Pond. My mom hates this picture. The fab site had significantly more phosphate and ammonia compared to non-bloom areas. So this just leads us to a huge question. We're left wondering if higher nutrient concentrations are influencing fab growth, or if fabs are just present in this area due to already available nutrients and they're just thriving off of those nutrients. So this could be a potential area of focus for further investigation if I had more time. And then looking at POM data, our fab site had over 100 times higher concentration of particulate organic matter compared to our non-fab site. However, we were not able to run a t-test between bloom sites and non-bloom sites due to small sample size, because if you guys remember, we only had one fab site and our other two sites did not have fabs. So my key takeaways would be that the fab site had a significantly higher nutrient concentration than the non-fab sites, but we do not know yet if fabs are causing or creating these shifts in chemical shifts. And overall, protecting mountain lakes starts with understanding these changes and what they could potentially mean for our future. For future work, I would like to look at nutrient sources as well as how fabs can impact macro and microinvertebrates in lakes, as well as how macro and microinvertebrates affect fabs. So just like how they intertwine, as well as seasonal changes. So does temperature affect it? Does wind speed affect fabs and their growth? So a special thank you to everyone coming to listen to my presentation. And thank you to Carla, Katie, Abby, Isabella, Alicia, and Madison for allowing me to take a huge step towards my goal. And these are my acknowledgments and some of my favorite pictures from the field.